you. My name is Shady Amin, and I'm an associate professor of biology here at NYU Abu Dhabi. My lab is called the Marine Microbiomics Lab, and so we study host microbiome interactions, focusing a lot on the marine environment. So we study phytoplankton, harmful algal bloom microbiomes, as well as corals. But we also do a little bit of work in the terrestrial environment, for example, in the human gut microbiome. So today I'm going to talk to you about a story that's currently in revision, so it hasn't been published yet. And it takes us a bit from genetic adaptation into microbiome-mediated adaptation. And so I'm going to talk to you about this group, this genus of bacteria called Endozoicomonas, which are believed to be coral symbionts, so they exist inside the tissue of corals. People have seen them with corals regardless of species, regardless of geography worldwide. However, we don't really understand why they're symbiotic. Okay? So coral reefs are obviously very important. They support about 25% of global fisheries. They play an important role in protecting coastal zones from erosion. And they obviously have very important economic as well as touristic advantages to the coastal areas that they exist in. But because of global warming and climate change in general, coral reefs and corals are really struggling to survive. And because of lots of bleaching and coral diseases that are infecting these corals, they're really facing the threat of extinction. So corals here in the Persian/Arabian Gulf are also exposed to obviously a lot of very extreme environments. And so I'm showing you here the sea surface temperature in the ocean in general, going basically from summer until the end of the year. And what you see is the Arabian Gulf, situated right here where we are when we started in August, is really the hottest body of water on Earth. With a mean temperature of about 34.5 degrees, very different from what you'd find in the Great Barrier Reef, where we have the biggest coral populations in the world at 29 degrees Celsius. Corals here are exposed to a very high maximum temperature that exceeds 36 degrees. There's also huge seasonal fluctuation between summer and winter, where the temperatures can drop down to 16 degrees Celsius, so it's a huge difference. And they're also exposed to high salinity, which is much higher than what is typical in the average rest of the ocean, as well as strong UV exposure. And so how do corals deal with all of these environmental stresses? They typically rely, one of the mechanisms that they rely on is the fact that they have a coral microbiome that's critical for their survival and is quite complex. And so here I'm showing you a coral polyp. This is kind of the living part of the animal. And around here you see depictions of members of these microbiome. The most studied ones are these algal symbionts. They belong to this family of algae called Symbiodinaceae. And they're essential for coral survival. So if you've heard of coral bleaching, that typically happens when these guys are not in the coral anymore and the coral starves and dies. We also have lots of bacteria. Some of these bacteria live kind of at the surface mucus layer that corals produce. Some are present inside the tissue, as these Endozoicomonas bacteria that I will be talking about today. And then we have a whole bunch of other microbiome members that vary from fungi and microeukaryotes to viruses and archaea. And really, for the most part, we don't really understand the majority of what these microbiome members are really doing with the corals. We know they're important, but we don't really know their functions. And in fact, most of the information that we have so far come from a relatively small number of experiments and a whole bunch of genomic data. So a lot of it is really putative, and that's basically what you see here. Most of these are based on genomic information. And it generally revolves around these microbiome members getting shelter and protection from the corals, from the corals, yeah. And then in response, they get a whole bunch of different things from that they give to the coral. So for example, the algal symbiont, because it's photosynthetic, it provides a lot of photosynthase and organic carbon that feeds the coral. We also have a lot of bacteria that do a lot of nutrient cycling, particularly sulfur and nitrogen, that is important for the corals. Some that produce antimicrobial activities that supposedly can fend off coral pathogens, as well as getting rid of reactive oxygen species. And we know that when this microbiome kind of undergoes dysbiosis and it kind of starts to fall apart because of these extreme environmental stresses, that we end up developing a variety of coral diseases that end up basically killing these corals. So Our ultimate goal and objective is to really shed light on these mostly unknown coral microbiome interactions and how these enable the corals to withstand effects of climate change and particularly global warming. And we're hoping to really, kind of a long-term goal is to use corals in the Arabian Gulf, given that they're adapted to this extremely high temperature tolerance, that we can actually use them to figure out a method for developing probiotics that we can then use to potentially save corals elsewhere. And for today, most of what I'm going to be talking about is going to be using combined field sampling from corals across a temperature gradient with multi-omics and specific lab experiments to look at some of the patterns that we see in multi-omics to elucidate some of these interactions. So our kind of scheme of sampling looks like this. So this is basically just the annual temperature patterns. And we've sampled corals at 3 different temperatures. So at 32 degrees, which is considered to be kind of the beginning of the stress, the onset of stress in June. At 34 degrees in the middle of the summer, about August, where corals are extremely stressed. And at 27 degrees in November, when the temperature drops, and thus corals are kind of recovering. We've sampled 1 specific species of coral, which is shown right here, Acropora pharaonis. And what we do basically is you take small pieces of the coral, and in this case from each of those temperatures, at 34 degrees specifically, where the temperature is very high and corals are stressed, some of the coral colonies developed a specific disease called white syndrome, which you could see right here. And in that case, we've also collected samples from 2 parts of that diseased colony, from the kind of the whitish part, which we call lesion or L, and the healthy part that's right next to it, which we call adjacent to the lesion or AL. We take these pieces of corals, put them on top of a ship, so these are collected by scuba, and then we extract DNA from those for shotgun metagenomics, as well as metabolites, which we use mass spectrometry untargeted metabolomics to look at what molecules are there. And when we look at the metagenomes, I'll start with that first. One of the problems you always run into when you're working with host and microbiome, especially if you cannot separate the host cells from the microbiome, is most of your reads come back as host. And so what we have here is roughly 75% of our reads are coral reads. And in our case, corals are even more complicated because they have this algal symbiont that's highly abundant. And so another 20% of the reads belong to that symbiont. And so we're working really with the microbiome of only 7% of the read. And so in order to overcome that problem, you have to basically oversequence. This is the microbial composition that we see in the coral. Not important really to see what's in there. We can look at the relationship between these samples in this coordinate analysis plot. And so what you see is generally there is not much differentiation between the coral samples at the different temperatures. So 27, 32, and 34 degrees all cluster right here. We do see a bit of separation between the diseased samples, the L and AL right here, relative to the healthy samples. But when we go to the metabolome, we actually see much more finer resolution than we see with metagenomes. And so the 27 and 32 degrees all cluster right here, which actually makes sense because right at the onset of stress and at the recovery stage, those 2 states should be more similar to each other than at the middle of the stress. And that's exactly what we see. 34 degree samples, most of them lie right here, pretty different from 32 and 27. And they cluster a little bit closer to the disease samples. So what we wanted to then, from going from this kind of overarching look at the metagenome and the metabolome, we wanted to look at the functions of the members of the coral microbiome that are doing this stuff. And so here I'm showing you basically the most abundant KEGG pathways that exist in those metagenomes, both from coral symbiont as well as the microbiome. So if the color is cyan here for each one of those pathways, that's coming from the microbial component. If it's green, it's from the algal symbiont. Brown is coral, and gray is shared between any of those members. And what you see, which is typical, is a lot of shared pathways. And that's common, right? There's a lot of biochemical pathways that are shared among all life. But what was really cool is we see very specific patterns here where, for example, steroid hormone biosynthesis is mainly dominated by the coral, while steroid degradation is mainly dominated by the microbes. And this was cool. This has not been shown before. So when we take our metabolomics data, and here we annotate this data, and then we can connect the metabolites to the genes that are link to them, and then we can do a similar type of analysis. And we see the same exact pattern, a lot of shared metabolites, even more so than the genes. But we see again that the coral, right here, is dominating the steroid biosynthesis, and steroid degradation is dominated by microbes again. So this is great, but it of course doesn't really get us into the nitty-gritty details of what's happening, especially with that microbiome. So one of the typical things that we do with metagenomes is you assemble genomes from there. And when you assemble genomes, then you have specific microbial groups that then you can look at and see what potential things they might be doing. So we call this metagenomically assembled genome. So you'll hear me say MAGs a lot. That's what it stands for. And see, these are the MAGs that we've assembled from our coral metagenome, colored here by the different families that they belong to. And the one that I really want to point your attention to is down here, which is called MAG10. So this is an Endozoicomonas MAG that we were able to recover, which was really nice. We can also look at the relative abundance of these genomes in the metagenome, and you do this by read recruitment. So you basically, you have your genomes and you have your metagenomic reads. You can align your metagenomic reads to the genomes and quantify how much of them are aligned. And you basically get something that looks like this. So we have here our MAGs. Whoa, 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 whoa. Okay, sorry. So we have our MAGs, and right here, MAG10, this is the Endozoicomonas. You see that a lot of them are not really doing much across the different samples. Some of them seem to be potentially opportunistic because they increase in abundance during disease. But we see the Endozoicomonas, which is supposed to be symbiotic, is highly abundant in healthy corals, and then they collapse in numbers when we get to diseased corals. And this is consistent with people, work from other people. So this was nice to see. So then we were really interested in this on the Endozoicomonas and what potentially could it be doing to the coral. So we've done a lot of functional enrichment analyses and a lot of genome mining that I'm not going to bore you with all the details of that. But I'm going to try to represent all of this in this very big complex figure, which is basically a pangenome of Endozoicomonas genomes that are available publicly in addition to our MAG10 that we recovered from this data set. So right here, the heat map that you see up there is basically the relationship between each of those genomes. It basically shows the average nucleotide identity between each pair of genomes. So the darker the color, the more similar those genomes are at the nucleotide level. And MAG10 is right here at the bottom, and it clusters very nicely with a bunch of Endozoicomonas acropori genomes. So those are Endozoicomonas that were isolated from acropora corals. Same was the case with ours, so it's nice that it clusters with those. Here you see a whole bunch of biosynthetic gene clusters and their counts in the different genomes. And then you have a whole bunch of tracks that go around here. Each one of those tracks represents a single Endozoicomonas genome. And the color of the track represents the origin from which this genome came from. So if it's blue, it came from a hard coral, like the acropora that we've sampled. If it's dark blue, like these two, that came from a soft coral. Orange are sponges, gray are other invertebrates, and the one red track that you see right here is the closest free-living relative to Endozoicomonas, because Endozoicomonas are all intracellular with corals. And when you see, so basically each one of those tracks is made up of genes, right? Each gene is a thin line, so you have thousands of genes lined up. If the color is dark, that means that gene is present in that genome. If the color is opaque, as in this area, that means that gene is absent from that genome. And when we cluster these genomes by gene function, what you see basically is there's a whole bunch of core genes that are present in more or less all of those genomes. There are accessory genes that are present in some groups and not others. And then there's a whole bunch of singletons that are unique to each one of those genomes. And we found a lot of really interesting function from the functional enrichment. I want to just tell you a story about two of them only. One is this one right here, KSTD, which is related to the steroid degradation, which I will tell you about at the end of the talk. And the second one is this gene right here, IUCC, which is actually the canonical gene for a biosynthesis of a molecule that belongs to a family called siderophores named Aerobacter. So why is this interesting? So to tell you about siderophores, which is a Greek word for iron-loving, I'll have to tell you about iron. So iron is required for almost all life on Earth, but one of the problems that it has very poor solubility in aerobic environments. And in addition to that, whenever it exists as in a soluble form, iron is always bound by organic molecules of unknown origin. Particularly, we think they're microbial, but we don't really know. And so when you look at the concentrations of iron, for example, in the ocean, it's extremely low. It's in the picomolar range. And so because of that, iron is the major limiting micronutrient, both in the oceans, in soil, as well as actually inside animal hosts. So, and this may come as a surprise, but in our blood, right, the dominant reason why our blood is red is because it has iron bound to heme. But believe it or not, when you have a pathogenic bacterium infecting us, these bacteria cannot survive inside our body unless they have a way to steal this iron from our red blood cells, which is extremely difficult. And so a lot of bacteria, or not a lot, but some bacteria have evolved mechanisms to deal with this by secreting these small organic molecules that have extremely high affinity for iron, known as siderophores. And so there are hundreds of siderophores that have been characterized, the majority of them really from pathogenic bacteria like E. coli and bacilli and so forth. Aerobactin is right here. And the way siderophores work is basically bacteria synthesize the molecule intracellularly, and then the molecule is exported outside. It binds iron near the cell surface. And then basically you have a dedicated receptor that binds that specific siderophore iron complex, and then that helps the iron get transported inside the cell where it gets chaperoned by whichever protein into its final location. But there was one thing that didn't really make sense. First of all, aerobactin is produced by several pathogenic enterobacteria that use it to basically steal iron from our human proteins. And in general, inside animals, it's unlikely to see any bacteria that make siderophores unless they tend to be pathogenic bacteria. So if Endozoicomonas is a symbiotic bacteria, it makes no sense for it to be stealing iron from its host, right? And so what we've done is to try to look more carefully into this, and this actually turned out to be a very good lesson in not relying on gene annotation alone to infer function. So here we're looking at the gene neighborhood of this aerobactin biosynthesis gene cluster across these Endozoicomonas genome. I'm showing you, let's start right here. This is Klebsiella pneumoniae. So this is a pathogenic bacterium that makes aerobactin. And the IUCC gene is this yellow gene that you see right here. It has two copies because aerobactin has symmetrical two-fold symmetry, so it needs two copies of the same gene to make those two arms. And it has two other genes right here that are part of that biosynthetic gene cluster. And right next to it, you see this receptor. That receptor is the one that takes up that siderophore once it bound iron and gets it inside the cell. And what you see is, when you look at the closest free-living relative of Endozoicomonas, it has the same thing. It has all four genes. It has a receptor, fantastic. When we look at all the Endozoicomonas genomes that I'm showing you right here, every single one of them have the biosynthetic genes of aerobactin, but not a single one has a transporter. And if you don't have the transporter, you don't have a functional siderophore uptake system. And so we figured, well, perhaps what's happening here is Endozoicomonas moved into the coral, and basically they're acclimating to this kind of endosymbiotic lifestyle, and they're starting to lose the ability to produce a siderophore, and the first thing they lost is a transporter. So to confirm that further, we took the protein sequence of IUCCs, and we basically looked at just the basic alignment. And what we see is all the Endozoicomonas genomes have more or less semi-similar sequences of this protein. With the closest free-living relative, you see that the sequence is divergent completely. And so it could be an indication that there's some kind of genetic drift. But the ultimate proof is really to show phenotypically that this is what's happening. We cannot see aerobactin by mass spectrometry, at least when we try to test it. So that's OK. Could be that it's not being produced, but it also could be that we cannot detect it for whatever reason. But there's a very nice colorimetric assay to detect siderophore called the CAS assay, or CAS stands for chrome azrol S. It's basically a dye that weakly binds iron and is blue in that state. If you add a siderophore, it extracts iron from the dye, and the dye then changes color from blue to orange. And so that's what I'm showing you right here. This is our positive control with just EDTA, which is a very good iron chelator. Negative control with just water. And then whether we take the whole culture of an isolate of Endozoicomonas that we got, or the cell pellet, or the supernatant, in all cases, we don't see any evidence of siderophore. So this kind of tells us that there is most likely a loss of the function of producing siderophores by this bacteria, mainly because of its movement from a free-living organism into an endosymbiont, into a coral, where it does not need to steal iron anymore from the host. The second thing I want to talk about relatively briefly here, is this other gene, KSTD, which is responsible for steroid degradation. And so what I'm going to show you here is kind of like a map of all the steroid biosynthesis and degradation that we see based on both metagenomic and metabolomic data. So here in this circus plot, we have metabolites in black, and we have these colors represent different pathways related to steroids. And what you basically see is Endozoicomonas is the only member that's able to degrade steroids. There's nothing else in that microbiome that's able to do that. The coral host dominates the biosynthesis of both steroids and steroid hormones, and there's a little bit of contribution of steroid biosynthesis from the algal symbiont. Okay? And what we can do then is we can look at both our metagenomic and metabolomic data and map out what happens with those in the coral. And so in the coral, we know that it takes cholesterol, and it basically passes it through multiple pathways or multiple reactions in order to get to these terminal steroids, progesterone, estradiol, and testosterone. And what we see with the bacterium is that it has this really nice KSTD gene that enables it to take testosterone and break it down into androstenedione, and then from there all the way down to the TCA cycle. So basically use it as a carbon source. But what was really cool when we looked at this that really surprised us is when we look at the relative abundance of these molecules in almost every case. And I'm only showing you some of those steroids. We have a whole bunch that just do not fit on this graph. You generally see that the relative abundance is high at 27 and 32 degrees, and then it drops at 34 degrees and in the ZZ samples. And that kind of makes sense, right? When the coral is happy, it's making its steroids, it's regulating whatever function it needs to regulate. And then when it's under stress, then you gotta divert this energy into something else. But what was really surprising, and when we look at these terminal steroids specifically, progesterone, estradiol, and testosterone, those were almost the only three molecules for which the relative abundance was highest at 34 degrees, even though none of the precursors that make these molecules are actually supposedly present. And so that drove us to think that perhaps what's happening, given that Endozoicomonas is able to do this transformation with testosterone, that perhaps some of these enzymes can work reversibly. And so they can actually provide these molecules, and that's why we would see their elevated amounts at 34 degrees when Endozoicomonas is actually abundant in the coral. So to prove this, basically we took Endozoicomonas in the lab, we grew it in the presence of another abundant steroid in the coral, which is cholesterol. So there's a lot of it inside the coral. And we can map out a potential pathway of biosynthesis from which we go from cholesterol all the way to testosterone as well as progesterone. And we see that with statistical significance relative to controls. And we can also see that some of it is getting siphoned by the bacterium into the TCA cycle, again, to use as a carbon source. And we can prove that with mass spec and fragmentation. And so this was really exciting because while many bacteria can take testosterone and can metabolize it, very few bacteria can take testosterone and make these very terminal steroids that really don't have any functional use in bacteria. So these are purely eukaryotic signals that are important for eukaryotes. And so what we think is happening here is that Endozoicomonas, when it's supposed to be free living, it has a whole bunch of biosynthetic gene clusters that enable it to deal with this environment. There's a whole bunch of those. But once it becomes endosymbiotic with corals, it doesn't need a lot of those. So it loses a whole bunch of those biosynthetic gene clusters, including aerobactin. It also gains a lot of interesting functions like steroid degradation and the production of some vitamins that may be useful for corals. And then we think that under thermal stress, when the coral is unable to make things like testosterone and progesterone, that Endozoicomonas switches from relying on testosterone as a carbon source to basically switching to cholesterol that it then potentially uses to make these molecules that then prime all kinds of different functions in the coral, including activating innate immunity. And with this, I think I'm running out of time, so I'm going to just skip here. And I'd like to just thank Amin and Michael, who really drove most of this project, as well as John and Trent, whom we have a collaboration on corals that hopefully will have a lot more exciting results than even this one. And thank you.